It's now my pleasure to introduce Dr. Rudolph Jaenisch, the recipient of the J. Allen Taylor Prize for 2011. Dr. Jaenisch received his MD from the University of Munich in 1967. He then performed postdoctoral research at the Max Planck Institute for Biochemistry and in the Department of Biochemistry at Princeton University. After a brief visit as a fellow in the lab of Beatrice Mintz at the Fox Chase Institute for Cancer Research, he was appointed as an assistant research professor at the Salk Institute. In 1984, he became a founding member of the Whitehead Institute, where he currently holds the position of professor of biology at MIT. Dr. Jaenisch is a pioneer in making transgenic mice. He produced the first transgenic animals by introducing viral DNA into mouse blastocysts. He went on to show that those retroviruses could be used to introduce foreign DNA into mouse embryos and that these mice could transmit these transgenes to the next generation. Dr. Jaenisch has used embryonic stem cell technologies to elucidate the roles of many important genes in various areas of biology. To name a few: P75 nerve growth factor in peripheral nervous system, MIF5 in muscle development, Wilms tumor gene in kidney development, BDNF in nervous system development, activin and inhibin in eye development and reproduction, exist in exon activation, and TIMP1 in tumor cell invasion. More recently, Dr. Jaenisch's laboratory has turned its attention to studying somatic cell nuclear transfer, and they demonstrated that the epigenetic marks on the inactive X chromosome of donor cells could be erased when transferred into an egg. This work supported the growing idea that the developmental states of cells, as recorded in the nucleus, could be reversed in embryonic or pluripotent cells. These studies of the molecular mechanisms involved in reprogramming of somatic nuclei generated the environment for Yamanaka's experiment supporting that the transient expression of 4 transcription factors was sufficient to generate a pluripotent cell from a differentiated cell. Those are iPS cells. Using mouse models of sickle cell anemia, Dr. Jaenisch has gone on to show that a combination of gene therapy and autologous iPS cells can be used to treat disease. Dr. Jaenisch has published over 400 articles and reviews. He has many, many awards, including the Boehringer Mannheim Molecular Bioanalytics Prize in 1997, the first Gruber Prize in Genetics in 2001, the Koch Prize in 2002, the Bubacher Foundation Cancer Research Prize in 2003, the Max Delbruck Medal in 2006, the Velsek Prize in 2007, the Massey Prize in 2008, the Ernst Schering Prize in 2009, the Kazemi Prize of the Royan Institute in 2009, and the MGH Warren Triennial Prize and the Wolf Prize in Medicine in 2011. He is also a member of the National Academy of Science and a fellow of the American Academy of Arts and Sciences. Today, Dr. Jaenisch will be talking to us about stem cells, the molecular control of pluripotency, and the promise for personalized medicine. Dr. Jaenisch.
Well, thank you very much for this really generous introduction. And I must say I am extremely honored to get the Taylor Prize. It is for basic and clinical research, and as you said, I have an MD. But I must say that I studied medicine in the 60s in Germany, and I really didn't like it. I really hated it. The reason was it was overcrowded. One couldn't get in any lecture room. I decided not to do this, and I escaped doing some work in phages, phage DNA replication. But doing this was very clear medicine was not a good basis for this. In Germany, when you say you studied medicine, oh, you only have an MD. It was really not very highly regarded there for good reason. So I thought maybe I need a PhD, but it didn't exist. Chemistry, it didn't exist. MD, PhD didn't exist in Germany. They just had started a biochemistry program in Tübingen. And so with a friend, we went to the personnel chief of Bayer, the biggest pharmaceutical industry, and asked this personnel chief whether he would be interested in having a double degree, somewhere an MD plus degree in biochemistry. And the guy said, oh no, he was very clear. He said, no, no, no. We need MDs to sell our stuff and chemists to cook it, but we have no. So that was a very clear answer, and I think it reflected the philosophy of the German pharmaceutical industry. And so it saved me certainly 5 years of work. So, but I have been really involved in stem cell research over the last 10 or 15 years. And I must say you can't really deny your medical training. And it really took me a while to realize that you have just a very different view of things, and you just have a different point of view when you do experiments. So I began to really appreciate that I got this, particularly with the IPS technology, where maybe I think this will indeed change at some point how we do medicine today. So I'm extremely thankful and honored for having this award to the Robarts Institute, to the Stiller family, and so I can't really say how much I'm honored by this. So what I would like to do is to talk about embryonic stem cells and what we have learned over the last 10 years, 15 years, about the promise and problems and what the reality now is. So as we had heard already, let me just make the point, when we know the sequence of one cell of the gene, and we know the sequence of all cells, but of course it doesn't help us to understand, because DNA genes are packaged into chromatin. We have as many epigenomes as we have cell types. So the key question of the field really is what distinguishes cell A from cell B, and how do you convert those to one another? I think this is sort of the major issue. And of course, the promise of stem cells is you can make any cell type of the body in the culture dish. So what's the goal? The final goal would be to use the potential of pluripotent cells to provide matched cells for customized tissue repair of some diseases. Now you can ask the question, does it work? And when you go to the internet, yes, it does. You see, for example, for good reason I do this. I'll give you some examples from Europe. This is a clinic in Düsseldorf in Germany, and they treated thousands of patients. One of the problems was some of these patients died. And it took the German authorities 1.5 years to close them down. It just occurred this year. The reason, I think, is because they used their own cells, and this is not a drug. So I think this is legally not a disease. But it was really, I thought, to my opinion, very embarrassing that something like this would survive in Germany for such a long time. And of course, there are people who do this very imaginative, like they're using embryonic stem cell culture from blue shark and put this directly into the injury site. So it's rather interesting. I'll give you another example. This company, also from Europe, they use as a measure of success this guy, Mr. Fernandez, who was as old as Bill Clinton when he was transplanted, and look at his hair and how much younger he looks as so it is really impressive. And my final example of this is from Switzerland, where they treat everything from impotence to burnout syndrome, anything, and they're very imaginative because they use a Swiss apple stem cell serum, which is derived from a rare Swiss apple, the Utwiler Spätlauber. So, of course, when you go there, you pay. It's $10,000 for 3 days of Swiss apple stem cell serum. You don't pay for the view of the lake. Actually, this is very serious. These are all over the world, not only in Europe, but particularly also in other countries. And people are desperate and go there. And for example, this is just one example of an email I got from a woman who went through several failed spinal cord operations and was in incurable pain. And this was the only hope she had. And of course, if you tell her these are charlatans and criminals who offer this, it doesn't help, because you find on the internet these movies where the patient lies flat, right, and now he walks. And how could this be wrong? So I think it's a major problem. So what I learned, what probably many of you learned, development is unidirectional. Of course, what Dolly told us, you can reverse it, and this said that nuclei of differentiated cells maintain the potential to generate an organism. you're putting then embryonic stem cells and nuclear transfer together, you come to the concept of therapeutic cloning, which was in the 90s a very controversial concept, much discussed. And we did now 10 years ago, so the concept would be use your own cells. And so the big promise was, there were big promises, but would it be really useful for therapy? Could you use this for any therapy? And so this was a proof of principle experiment done together with George Daly 10 years ago. We've used as a patient this RAC2 mutant mouse, which has no peripheral B and T cell, did nuclear transfer, generated a cloned blastocyst, then cloned ES cells, which will be customized to this patient here, and you would correct one of the two alleles by gene targeting, differentiate these cells to bone marrow stem cells, put them back into the mouse, and they restore the immune system. So this therapeutic cloning worked, but of course, if you have a human here, this is not an option, because it doesn't work in humans so far. So the only option was to do it without eggs, do directly, and of course this was then the birth of the iPS cells, 5 years ago by Shinya Yamanaka. And I think this discovery really have changed the field and have just started an explosion to many of the field. So it was based, Shinya's discovery was based upon knowing what the transcriptional regulatory circuitries of stem cells and these three transcription factors, as you all know, NANOX, OX2, and OX4 are very upstream of many downstream genes, and they also interact with each other. So they form this regulatory loop. So if I just summarize the Yamanaka approach with one slide, it was taking a cell growing in a culture, transducing with viruses these four factors, so the two pluripotency factor and two oncogenes, KLA4 and CMYC, and getting a mouse, which is quite amazing. So this whole approach, I think, is as amazing as nuclear transfer in the first place. And of course, it has, as I said, launched a whole new field. So we were interested in studying mechanisms. And of course, studying mechanisms, there was a complication because you used viruses. And that made it, the whole approach, very difficult to reproduce. It was reproducible, but very inefficient for reasons which I don't want to go into. So a key experimental tool for us was to do it without viruses. And so let me just briefly go through this because I'm going to use this system. We made this reprogrammable mouse where fibroblasts, which had a GFP marker in the NANOC gene, so they were NANOC negative, and infect those with doxycycline-inducible lentivirus, which transduce the four factors, and generate what we call primary iPS cells under doxycycline. Then we take doxycycline away, inject them into blastocysts to make a chimera. So the key is now the chimeric cells are derived from one of these clones. And then you can take any cell type you want from this mouse, explant those, give doxycycline, and what you get now is from these secondary somatic cells, you get secondary iPS cells. So the key is this goes without any viruses because you pre-selected for the right viral combination in the first place. So this made it much more efficient. So it was about 50 to 100-fold more efficient than if you use a de novo viral infection. So using this system, we asked, I just summarize what we found was about the direct reprogramming, how we understand it, was the key is reestablishing the core autoregulatory loop. And we know every somatic cell has a potential to make iPS daughters. And it was done with B cells where we had a correct marker. The process initiated by the four factors, they're not a switch. They initiate a long process which involves extensive epigenetic resetting and multiple cell divisions. So that's important. So the key is to re-establish this loop, and then all the downstream genes will fall into place under the right selective conditions. So let me come to to what we think people want to use the system for. And this is so if you have a patient with some disease you want to study, then of course you would make these iPS cells from skin cells or from blood cells, the various sources. And if the patient suffers of a neural disease, then you would have to know differentiate these cells in culture into neurons and study this. If it was a liver cell or beta cell disease, then you would do this to the respective cell type. So the goal, therefore, I think, of human stem cell and iPS research would be to study human diseases in the Petri dish. I think this is the lowest hanging fruit. I think this is realistic. And I will come to the issues. And possibly use it to find a drug which would help to treat a disease and maybe further way for customized cell therapy. I think that's not around the corner. But there are complicating issues which I really want to address. And these are human ES cells are not mouse ES cells. In most ES cells you can do anything. It's trivial to make knockouts, to make anything. With human cells you cannot, because they grow slow, they hate to be single, so you can't clone them easily. They need to be passaged mechanically, otherwise you select for chromosomal operation, and this is very, very labor intensive, and they have a very low efficiency of homologous recombination, which I think again has inhibited progress substantially. Actually, the question is, which I want to come to, what is a good ES cell? Right? So let me go to this. Or good iPS. Do we know what's good in human? In most, we know that. So human ES cells, of course, are derived from the Thomson protocol quite a while ago. It's a very robust protocol which everybody can reproduce. It uses FGF and activin, but not LIF. LIF is used for mouse. So the question I want to ask is the ground state of pluripotency, which I will define in a minute, is that realized in these cells, in the conventional cells, efficient protocol established by Jamie Thomson? So you saw a picture before of this. So we can make pluripotent cells from various stages of the embryo. From the inner cell mass, these are the classical mouse embryonic stem cells, or from the implanted embryo, from the epiblast, and these are called epi-SC cells. Both of those are pluripotent, but actually they're very different. So let me come to the differences, and then come to the human system. So in the mouse, we can derive ES cells from the blastocyst, or iPS cells from somatic cells, or epi-SC cells, the epiblast stem cells, from the implanted epiblast. In human, we can make ES cells from the blastocyst and iPS cells from somatic cells. But these cells are very different. So most cells are small colonies, small cells, compact. These are flat colonies, a typical human ES cell morphology. They have different growth requirements. Live start for most ES cells, basic FGF and activin for the human. Gene targeting is very efficient here, very inefficient in both of these cell types. Exon activation. The most cells are pre-exon activation. Human ES cells and epi-SC cells are post-exon activation. They already have undergone exon activation. And Austin Smith has really named this state the naive or grown state of pluripotency, as characterized by the classical mouse ES cells. And this one deprimed, or the epiblast-like state for human and the epi-SC cells. So implying that these are less immature than the naive cells, for example, as seen by exon activation. And I want to come a little bit to exon activation. So there are different states. There's a naive state, which is unlike the primed state, which is the one of epi-SC cells in the mouse and human ES cells. So can you convert those? So are these, clearly they're stable states. Can you convert them? And in these you can. Naive cells can be converted to the primed by exposure to FGF and activin and some other things. And actually the primed cells can be converted to that state by LIF and 2I conditions, which I will come to. It's from Austin Smith. So this is really based, much of this, on Austin Smith's work. So when we tried this in human, it just didn't work. Right? So then I had a very great postdoc, Jakob Hanna, and he used, there he is, Jakob Hanna together with Chris R. He used a human secondary system. Now it was very close what we did in the mouse. So in this case we used human primary fibroblasts, infected them with doxycycline-inducible viruses. then made these primary iPS cells after DOX withdrawal. We would differentiate those in vivo and vitro, and then generate genetically homogeneous secondary fibroblast lines. So again, they had the right combination of viruses, which allow, just for doxycycline to make secondary human iPS cells. So again, there was no need for viruses. So it was efficient. So Jakob used that system. So used these secondary fibroblasts, which carried these 4 factors, gave doxycycline to activate them, and then used these 2 eye conditions, the ERK inhibitor and the GSK3 beta inhibitor, so activating the Wnt pathway, and live. And asked the question, what happens to these cells? And what happened was rather surprising. So you can see here the flat cells you start with, and then actually a few days after exposure to these conditions, you find cells change, and then after many cell divisions, you find these tight colonies which really look like most ES cells. So what are these cells? Well, I come to the exist, just coming back to exon activation. Exon activation, as you know, starts with activation of the exist gene, which codes then, this non-coding RNA codes the inactive X. And you can see this by FISH in the undifferentiated cells. You see both axes, which are active, to express a low level of exist, but then after differentiation, you see the bar body here coded by the exist RNA, and the active X has turned off exist. Now in human cells, you start here. Right? It doesn't change when you differentiate. You never see this state. So what happens in these? So you can see so we can either conventional human ES cells or these secondary cells, and you can see every cell expresses exist. But now when you go through this differentiation process, none of the cells expresses exist. But if you differentiate these cells, then all cells randomly inactivate and re-express exist. So they appear to look like most ES cells. And then we did many other experiments, tell you they cluster by gene expression profiling. The naive converted cells cluster very different from the primed human ES cells, and they actually cluster together with the most cells and very different than conventional human ES cells and the APSC cells. So they appear to really correspond to the more the naive state by many other criteria, which I don't want to go into. Now, why is that of interest? So what's the difference? So let me just, in the most we know this very well, the naive cells have a high proliferation rate, they have high cloning, single cell cloning efficiency, very low here, they have a high gene targeting efficiency, they have pre-exon activation, post-exon activation. Those cells can make chimeras, those cannot. So if you look now in these conventional human ES cells and these converted naive cells, they have many of these characteristics similar. They grow faster, they have a high cloning efficiency, as high as those, and you can passage them in trypsin. They do not acquire chromosomal aberrations. We don't know about gene targeting. They have reactivated the X, as I showed you, and of course we don't know about chimera formation. So they look like naive cells by these criteria we have, like most naive cells. And really the question now is, working with these cells, if we can make robust protocols to keep them, at the moment we need transgene expression, would this facilitate gene targeting? Would it facilitate to grow the cells and differentiate and so on and so forth? These are issues which you are very interested in, and we're setting up to study that. But let me come back to the question, what's a good ES cell? So we know there have been much data between the difference between ES cells and ES cells. Is everybody who works with human ES cells knows cell A is different from cell B and their potential to differentiate and so on and so forth. There are major difference between ES cells and iPS cells and between iPS cells and iPS cells. And these are just some of the papers which have recently appeared which show genetic as well as epigenetic differences. So the key is the major difference between ES cells and iPS cells. What could this be due to? Well, it could be for the iPS cells particularly to vector integrations and expression, which is of course individual. And I will come to this, will be a key point I will make later. It's individual for each iPS cell. You don't reproduce this. This could lead to different stoichiometry of factors. Genetic background of the cells is different. And of course, it's conditions how they were derived. I really want to come to this. And it was based upon an experiment which was published last year from Konrad Rehlinger, who used to be a student in my laboratory and has his own lab at MGH. It was a very interesting paper. What he showed was that the silencing of this imprinted locus, DLK1DR3, occurred in the great, great majority of all iPS cells he analyzed. And this, of course, does not occur in ES cells. Loss of imprinting correlated with a significantly reduced potential to generate chimeras, and none of these loss of imprinting iPS cells could generate what's called all iPS mice by tetraploid complementation, which I will define in a minute, which is the most stringent test for pluripotency. So the key conclusion was there was a strong correlation of loss of imprinting at this locus with reduced pluripotency. And the data were really convincing because they were based on a very defined transgenic system. So let me just go through this. What he used was a mouse which has the 4 factors as a polycistronic vector inserted into the collagen locus, the 3' UTR, and this was activated, was DOX-inducible, from the transactivator inserted into the ROSA26 locus. So you can make any cell type from this. There will be, and we can just give doxycycline. You make, no, that's key, isogenic iPS cells. They're exactly, and then you can ask the question, what's their potential? You can make chimeras, or you can make these all iPS mice where you inject not in a diploid blastocyst, but in a tetraploid blastocyst. And the tetraploid cells cannot contribute to the embryo. They can make the placenta, so there's a complementation. And the mouse is now forced to only be composed of the injected iPS cells. That's what's, whereas here you, of course, have help from the host cells, which are from a normal embryo. So the key is these iPS lines are isogenic, they're identical, and they have an identical stoichiometry of factor expression. So this was a very convincing paper. Of course, this system, this inducible system, was developed when Konrad was in my laboratory. He had really very much worked on this, and of course he did these experiments, he took them with him, but another student in my laboratory did the same experiment, Bryce Carey. He just was too late. So I told him, as Bryce is a good paper, I think you should give up. You got scooped. But you can't, MIT students, you can't tell them this. So of course he continued, and what came out was rather interesting. So this is a comparison between 2 almost identical strains, because he used the identical system with just minor differences. So let me just go briefly. So this is the vector Konrad used. So it's inserted into the collagen locus. It's OCT4 separated from CALA4, the second one, by a 2A sequence, which comes from aftoviruses, which allows the ribosome just initiate at the next ATG with the same efficiency as the first one. And the last 2 factors were under the iris control, so really hops on, comes on again, SOX2, then again a 2A sequence, and then CMYC. Bryce made this vector, and this has 3, we thought, absolutely minor differences. One is the second and the third factor were different. The second one, the 2A sequence was different. Actually, Bryce rejected this one. He put it in his vector, he tested them all. And we didn't use an iris, but a 2A sequence. We thought actually the iris was a very clever idea because iris really makes lower expression of the downstream genes, which I think is a good thing. So I thought it was a superior vector. But anyway. So these were the 3 differences between these transgenes. Now what does Bryce found? So when he looks at GTL2 expression, which is the transcript from this imprinted locus, he finds that the majority of the cells, there was no loss of imprinting. Only about 30% lost it. And those, when you look by DNA methylation, you see those which retain it have a characteristic HAPA site which is unmethylated and a methylated one, and those which lost imprinting have erased this. So it was clearly loss of imprinting, but really only in 2/3 of iPS cells or Bryce retained imprinting. When you looked at the generation of chimeras, there was no difference between the cells which lost imprinting and the cells retained imprinting, very much in contrasting to Konrad's data. So there's no difference in pluripotency between those as tested by this assay. And finally, he did the tetraploid complementation assay, and they found that all his lines, regardless whether they lost imprinting, could make tetraploids going to birth. And you see the green bars are the ones which make it to birth. I mean, many, of course, die. That's typical for this assay. They couldn't initiate breathing because tetrapod contamination, in general, your lung is a bit immature, so often they cannot initiate breathing, but then most of them could initiate breathing, and you see with the blue bars. And this would be one of these pups that lost imprinting and was breathing fine. So a high fraction of these OSKM, I call them OSKM, iPS cells are FOR incompetent, including those which have lost imprinting. And the final difference we noticed was Conrad's mice really get tumors very rapidly. It's really difficult. We have them. He sent them to our lab, and he reported this. They get tumors very rapidly. It's difficult to breathe them. Ours never got tumors. So high tumor incidence and short survival of those in contrast to ours. So of course you ask, what's the difference? This is a major difference. So of course vector expression. So we looked at this. And since exactly the same system induces, there was no difference. So background is was the same in both strains, and the inducibility was the same. There was no difference in RNA expression. So these are the major differences. In Conrad's mice, normal imprinting in a very few, small fraction, 70% in ours. Tumor incidence high, low here. Vector expression is identical. So what is the basis for this striking phenotype? This was the only difference. Everything otherwise was absolutely identical. So then Bryce did the key experiment. He did a Western. And then he found, comparing these two mouse strains, that OCT4 was about 10-fold higher expressed in our mice. And you see there's a lot of unprocessed precursors from this 2A sequence. And KLA4 was probably 20-fold higher expressed. Whereas, counter to expectation, SOX2 and MYC were lower, about half. So this is the quantitation of this. So much higher expression of KLA4, much higher expression of OCT4, but lower expression of SOX2 and MYC. So the major difference in level of stoichiometry of protein expression, although the RNA was identical. It was high OCT4 and KLA4 in our mice, low OCT4 and KLA4, but higher MYC and OCT4 in Conrad's mice. So what this predict was, maybe the amount of OCT4 and KLA4 protein is really key. So the question was, could we, when we increase this, could we lower loss of imprinting? Right? It was a very simple experiment. We just took these iPS cells, and in this case it was 45% loss of imprinting. If you just add OCT4, no difference. Just add KLA4, no difference. Both, there was a 3-fold reduction. So more of both of these factors increased the, well, normalcy measured by loss of imprinting. So additional OCT4 and KLA4 reduces LOI at this locus. But of course the key thing would be, could we restore normalcy or higher pluripotency to the non-permissive strain of Conrad's mice? And so could we change the stoichiometry, because the only difference between these strains, by adding this to his cells? So we did that, and the experiment was in the following, starting with his cells, got these isogenic iPS cells, and as I said, 5% had normal imprinting. We add, with the virus, OCT4 and KLA4. So there's a primary iPS cells, you won't call them, they're overexpressed. And then what was the GTL2 expression level? Well, it went up to 20%, which is not that high. But of course, we don't know what, I mean, this is a random viral infection. Which one is a good one? Of course, we can't predict that. So the key was then, could we make it more uniform if we take one such a cell, going through the bottleneck, and make tertiary iPS cells? So would they be better? So the experiment is then, we were here, right? Get these cells which overexpress, 20%. We take one clone, which had normal imprinting, and now injected this back into blastocysts, make chimeras, isolate secondary mass from those, get now cells which are all clonally derived from this clone, and then inject them again into blastocysts, make tertiary ones, and ask the question, would they be more homogeneous? And the answer was yes. They went up to 60%. So the key here was, once you go through the bottleneck, have something more optimal in one of these clones, you can make this almost as good as in our mice. So this would be then, the stoichiometry, it appears to be high expression. The quality is high when you have higher OCT4, KLA4 expression. It's lower, suboptimal when it's lower here. Teratoma formation is both. Chimera grade is high here, but it's in general low here. For incompetence, it's high here, it's very low here. and they are retained mostly imprinting. So what can we conclude from those results? So it appears to be that the quality of iPS cells is really depending on the individual circumstances. So it depends clearly on genetic background and on conditions that are particularly for a given iPS, such as vector integrations. It was absolutely identical in both strains. Factor level expression, or which we like to say is, believe is, the factor stoichiometry in the initial phase of reprogramming. And that's the only thing which is different in these two strains. Right? So this is, having two defined strains to compare them, I think allowed us to make this conclusion, which you couldn't make with viral infection. So if you think a little bit more about what implications one should think about when we think about quality of iPS cells, then the question is, can we really define a generic iPS state? Or is it rather, does it depend on the, I would almost argue, idiosyncratic conditions of is isolation by using the conventional viral infection. And of course, you never can retrospectively know what you did. It's gone, it's history, it's hidden. So I think, so when you compare iPS cells with ES cells, you really compare something which is generic iPS, right? Or is it more complex? And of course, a key is now, can we, using this type of idea and optimize maybe the stoichiometry and make better iPS cells. So what do we want to use a system for? Of course, we want to use it for disease modeling and eventually for cell therapy. But to realize this, I think we have to really resolve a number of issues. And so I want to go up to two. One is gene targeting, and the other one, I think the most key one, is a disease-relevant in vitro phenotype. And I alluded to this. And I want to come to this, what is a meaningful control cell? That's a key issue, and I will try to develop this now. So, genetic manipulation of human ES and iPS cells. Of course, all you know, it's very inefficient. So we used really two different ways of trying to see whether we can use it. One is zinc finger nuclease, and the other one is talon nuclease. So let me briefly define what these things are. So the zinc finger nuclease approach is the following. You have zinc fingers which recognize certain sequences. They are fused to a FOC1 nuclease, and when two adjacently bind, the nuclease is dimerized and induce a double-strand break. And if you just incubate the cells, you get a mutation here. It's a very efficient way to make mutations. But if you transfect also a vector which has homology to the two ends, this can integrate at the break site. So does it work to manipulate cells? And I will just really briefly summarize in one slide. It's work by Dirk Hockemeyer and Frank Soldner. 3 genes we started with, and those were OCT4, it's expressed in ES cells, and basically it's 100% targeting. One clone was rearranged. The AAV-S1 locus, so this is the common integration site of the AAV virus. It's really an ubiquitously expressed locus. It's expressed in ES cells as well as in all somatic cells. It's a great locus to insert in transgene and get predictable expression of an shRNA, inducibility, whatever. We use it a lot. And its targeting is 50%. Actually, we use it, we can target both alleles, one with the transactivator, the other one with the dependent, DOX-dependent transgene. It works beautifully. And PITX3, which is a transcription factor expressed in the dopaminergic lineage, which is not expressed in the ES cell, was 10%. So zinc finger approach allows efficient targeting of expressed and silent genes, and we have done it now for quite a number of other genes. It works. There's one problem with this, and I will come to this now. So the talon. Talon is really an incredible system. It comes from plants, from plant pathogens. When they infect a plant cell, they want to turn on certain genes and turn off other genes. And they use it by DNA binding proteins, which are a 30 amino acid repeat recognizes a single base pair, and 2 amino acids are the key ones for the specificity. So what you can make then here is these constructs with, let's say, 12 of these 30 amino acid repeats, and they will then recognize 12 base pairs. And I will give you one example. For example, the AAV1 locus, which we use a lot, we use it for we want to compare talons were the zinc fingers. And so these were our zinc fingers, which the two bound here, and the side, the cleavage side was in the middle. And when you were the same with the AV talons, 18 base pairs were recognized. They bind a little bit further apart. You have to engineer it this way because they're more bulky, and the cleavage side is there. So what is the efficiency of these two systems? And I just want to give you this in one slide. It's actually totally identical. So the OCT4 was about 90% in both systems. The AV was 50%, and the PIDX3 was about 10 to 15%. So both of these are efficient, and we have done onto numerous other genes. We can insert GFP and I will not really go into this, but I will come to one more application. You need very short arms of homology, no need for isogenic vectors, and the off-target cleavage rate is very low in both of those. Now, why talons and not zinc fingers? Because you don't have to deal with sigma. You know, it's very difficult to design zinc fingers, and you can purchase them from Sigma, but it's very expensive. So I think the talons you can do in the lab. You do it in 3 to 5 days. You have your constructs you can start working with. So that's, I think, is a distinct advantage of the talon-based targeting approach. So what's the main challenge? So I think we have the number of papers have been published where they find difference between iPS cells from a patient or from, as opposed to controls, and I just give you a few examples here. And I think some of these were surprising to me, that you find a phenotype for schizophrenia, such a thing which is so complex, finding in a culture dish. So we have been interested in Parkinson for quite a while. Where we made the iPS cells and differentiated the dopaminergic neurons, there was just absolutely no difference. And I think this highlights the issues. So when you have a disease like Parkinson's, which develops over decades, and you have an iPS cells, you don't have decades. You have weeks or a month or two. And so the key issue, I think, of the field would be to learn how to differentiate these cells over the right precursors, and then likely to stress these cells with conditions which are thought to contribute to the disease, and maybe telescope aging. And then you could study, if you find a phenotype, you could study that. So that would be great. And so I think we're trying this, but so far there's not much to talk about. But there's one key challenge. I think that's for all of these experiments. And this is, what is the right control? Because you might, in Parkinson, you will have possibly a very subtle phenotype in differentiation. So is this due to you comparing two different iPS cells? They come from a different genetic background, from a different fibroblast, which was reprogrammed with a different stoichiometry. And I can go on and on. So the question is the phenotype due to the what you hope to study, to the disease, or to system-immanent variation? And of course, even a healthy sibling is not a good control. It's different fibroblast, and of course, it's not identical, and this is an identical twin. So the goal, I think, is for us at least, would be to make isogenic pairs of cells that differ exclusively at the point mutation at the nucleotide, which is disease relevant, which causes the disease. So the idea would be to either take an iPS cell, and it only works with monogenetic diseases, of course, and you correct the defect, or introduce a mutation into wild-type ES cells. So you can use, of course, several mutations. So the key is you make isogenic of pairs of cells. So when you differentiate those, you can compare apples with apples. So we are interested in Parkinson's, and there are many point mutations in the synuclein gene, which lead to early onset of Parkinson's. I will just mention A53T and E46K. And so the goal was to make pairs of cells which exclusively differ at this nucleotide, coming from the same embryo in case of ES cells, or from the same fibroblast in case of the iPS cells. So that was the idea, to either introduce these mutations into ES cells or correct those into an iPS cell. And we used various strategies. It's all based on zinc fingers at this point. One was vectors carrying the mutation. Of course, when you put the vector in, you can do this by strand migration, you have to cut out the vector again. because you can't leave it in, which leaves a footprint, a lock site. And it's a two-step procedure. Or what I really want to briefly mention is using a vector which has no selection, or actually an oligo, just an oligo of 30 base pairs, which has a mutation. So there you would leave no footprint. Of course, there's no selection, only screening. And so let me just briefly mention this here. So these are the two mutations, and we transfected wild-type ES cells with an oligo which either contains this mutation or this mutation. And then went through this, and you see here, actually this mutation was just at the cleavage site, the A53-2 mutation, and this one is about 21 base pairs upstream of this cleavage site. And actually it worked for both. So we got, it's not very efficient, it's a 1%, and you have to screen a lot. It's useful to engineer a restriction site into your mutant cells, so you can easier screen, but it worked. And these are now really cells which are absolutely, they leave no genetic imprint. So to our opinion, the oligo-based approach, although it's a lot of screening in the beginning, but once you have it, you have a cell which has really no lox P site left inside. So I think you have to match cells. And this, of course, if you find here even a subtle phenotype, if you find this in your pairs of cells, you might be confident that this might be disease relevant. So we try to do this approach now for different types of diseases. So what are the challenges for iPS cell disease research? I think you need vector-free iPS cells. Clearly, I think this is very clear how we do this now. It probably needs a little bit more efficient, but I didn't go into these technology. I think the quality control for pluripotency, what do we best use, naive versus primed. I didn't talk about oxygen tension. I think these are issues which are not resolved. What are your criteria for pluripotency? So in human, of course, that's difficult. Gene expression, teratoma, in vitro differentiation, none of those is terribly quantitative. I think we have to learn that. Efficient genetic manipulation, I think the zinc finger and the Talon approach will allow us to do this. And I think the key is to get these, to my opinion, get these isogenic cells. And then I think the key remains, and I think Mick emphasized this also this morning, really can we differentiate these cells sufficiently to the relevant cells? And I think that's the main, the main issue. Okay, so there were two applications. There were the study in the test tube, but then of course people think about transplantation, and it really hasn't been done yet in human, although this Geron trial was just aborted. So, but we thought, can we do a proof of principle experiment in mice? And I don't briefly in the last two or 3 minutes talk about the sickle cell anemia experiment. And so, in this case, as you know, sickle cell anemia is due to a mutation in the globin gene, and the erythrocytes sickle and lyse, and they clog up the small vessels, and you have infarct in the brain and the spleen, very severe anemia. It's a really very serious disease. And the model we used was a humanized model for sickle cell anemia, which was made by Alan Towns. And so what he did was he exchanged the most globin genes for the human versions of either the sickle version or the wild type. And these mice, when they are homozygous, they sickle. And they have these huge spleen infarcts. So actually, this model very well resembles the human disease. So the experiment was to take this anemic, miserably looking mouse, do a take skin cells, transduce them with these 4 factors. It was a while ago we did this, and got genetically identical iPS cells. We removed MYC. It was LOX. We didn't want to give these mice just leukemia, the activation of MYC. So it was cut out, and then we corrected the mutation, and then differentiated those 2 blood stem cells and put them back into the mouse, and you see this remarkable recovery. It was done again by Jakob Hanna. And just briefly tell you what the basis for this was. So these are experimental animals. They have a very low hemoglobin content compared to the control. Red blood count is less than 50%. Hematocrit is low. Retic count is high up because these bone marrows are under high steam to produce immature red blood cells, and the animals don't gain weight. But really after 8 or 12 weeks or later, this is almost normalized to 80 to 90%. So the hemoglobin goes up to 85%, red blood count goes up, hematocrit to 90%. The retic count goes really down substantially. There's some activity in the bone marrow still, and the animals gain weight. So I think this is really probably one of the most attractive systems for a cell therapy using this technology, because this step is really a routine in the clinic. But as we heard this morning, this step is a problem, right? So we don't know at this point how to differentiate again cells from human ES cells or iPS cells to something which can be transplanted. And I think that, again, this is the key issue to be resolved. So if one looks at this picture here, where you take from a patient these iPS cells, do all sorts of tests, and the idea would be we should be able to study the mechanism of disease in a different way than we could so far, and maybe find drugs and eventually do therapy. I think this looked 5 years ago or 6 years ago like really you write it in your grant, but you didn't believe it, right? Of course not. But now I think it's much more reality. I believe the iPS technology really has put this into something which is much more realistic. And I think the iPS technology not only, I think, told us how to reprogram cells and convert somatic cells to a pluripotent state, but really the transdifferentiation, where you go horizontally between cells, I think is based basically on this concept. And of course it works. And now suddenly transdifferentiation becomes a serious issue again. So I think it's really fantastic. It's a great time to work in this area. So let me come to an end here and just acknowledge the people who did the work. I acknowledge them in between: Frank Soldner, Dirk Hockemeyer, Hui Wang, Chris Saha. I mentioned Bryce Curry did all these, and Stella Marculaki did these experiments on stoichiometry, collaborated with Rick Young. Ola Isaacson, I didn't talk about the Parkinson experiments. Jakob Hanna and Chris Lengner were involved in these experiments. We collaborated with Sangamo very closely on the zinc fingers. And I think it should be only important that I also acknowledge the people who left my laboratory, but who were really involved in setting up the reprogramming field in my lab. Laurie Boyers now at MIT, Robert Bleloch at UCSF, Kevin Eck and Konrado Edinger and Alex Meissen were all students. They all ended up at Harvard. Katrin Platt is at UCLA, Marius Wernick in Stanford, Jakob Hanna just went to Weizmann, and Chris Lengner to UPenn. Thank you very much.
Okay, we'll take a few questions. Peter?
I think it's generic. So I believe there's nothing direct. So I think the loss of imprinting and this locus is probably a marker for low quality iPS cells. So I would, but it's certainly not causally involved. So that's something which I think it's really specific for the system used. And certainly in human cells, it's not seen. So it's not consistently seen. So it's not clear. What I think is, I can't explain why the stoichiometry, which is really transient in a way, why this would affect the state of these cells. I think it's a rather default, it's an epigenetic change which is rather general and just diminishes the quality of the iPS cells as tested by these assays. So to put your hand on what's going on might be not easy. So we have no idea what's behind it really. Yeah, so the tumor formation, I think, is rather, my explanation would be, so the Konrad's mice have this very high tumor incidence, and ours have none. I think it's just the vector. So they use an iris, which is driving MUC. And the iris, of course, can be activated from the collagen promoter by read-through. So you probably might get just occasionally MYC activation, which always gives you a tumor. Our MYC is strictly under the DOX-inducible promoter, which apparently doesn't occur. So to my opinion, it's a rather trivial difference between these two vectors, which reflects the tumor incidence.
No other questions, and let's thank Dr. Jaenisch.